Oxidase test. Welcome to microbiology class. What is the oxidase test? It's a rapid and crucial biochemical test used to identify bacteria that produce cytochrome C oxidase. Cytochrome C oxidase is an enzyme involved in the electron transport chain, allowing these bacteria to use oxygen as a terminal electron acceptor in respiration and produce energy. What happened in the oxidase test? When we add our colorless reagent, tetramethylphenylenediamine, to the bacteria. If the bacteria have cytochrome C oxidase, this enzyme removes electrons from the reagent. The electron is also transferred to oxygen in the air, and the reagent is oxidized and turns dark purple or dark blue. The oxidase test can be performed in 5 ways. 1. Filter paper method. 2. Oxidase disk method. 3. Swab method. 4. Slide method. 5. Direct plate method. Number 1. Filter paper method. Materials: Kovacs oxidase reagent 1%. Filter paper, sterile plastic or wooden loop, and fresh bacterial colony. In filter paper method, pour 1 or 2 drops of oxidase reagent onto the filter paper. Using a sterile platinum or wooden loop, pick a single colony from the fresh bacterial culture. Gently drag the colony onto the reagent-soaked part of the filter paper. Check the color change. The time required for the color change is up to 60 seconds. If color change to dark purple or dark blue within 10 to 30 seconds, test is positive. Or if color change to dark purple or dark blue within 30 to 60 seconds, test is weak or delayed positive. But no color change or color change after 60 seconds means test is negative. Number 2. Oxidase disk method. Materials: sterile distilled water, commercial oxidase discs, sterile plastic or wooden loop, and fresh bacterial colony. In oxidase disk method, place an oxidase disc on a clean, dry slide. If necessary, according to manufacturer's instructions, moisten the disc with a drop of sterile distilled water. Dry slide. Using a sterile wooden or plastic loops, pick up a colony of bacteria and gently rub it onto the disc. Observe for a dark purple color change within 10 to 30 seconds. When color change to dark purple or dark blue within 10 to 30 seconds, the test is positive, but no color change or color change after 30 seconds show negative test. Number 3. Swab method. Materials: Kovacs oxidase reagent 1%, sterile swab, and fresh bacterial colony. For swab method, first moisten a sterile swab with oxidase reagent, then touch a single colony of fresh bacterial culture with the moistened swab. Now observe the color change in the swab for up to 60 seconds. In positive test, develop it of a purple or blue color in the swab, but in negative test, there is no color change or color change after 60 seconds. Number 4. Slide method. Materials: Kovacs oxidase reagent 1%, clean and dry slide, sterile plastic or wooden loop, and fresh bacterial colony. In slide method, first place a drop of oxidase reagent on a clean and dry slide. Then, using a sterile platinum or wooden loop, remove a colony of bacteria. Mix colony in the drop of reagent on the slide. Now observe the color change to dark purple or dark blue within 10 to 60 seconds. In positive test, dark purple or dark blue color form within 10 to 60 seconds. But in negative test we have no color change. Number 5. Direct plate method. Materials: Kovacs oxidase reagent 1%, and fresh bacterial colony. Important points for performing this method. Do not use culture media containing fermentable carbohydrates, such as McConkey agar, due to the possibility of false negatives. Read results within the recommended time, as the oxidase reagent will oxidize over time when exposed to air after 60 seconds, causing false positives. Use a positive control, such as Pseudomonas aeruginosa, and a negative control, such as Escherichia coli, to ensure test accuracy. For direct plate method, pour 1 or 2 drops of oxidase reagent directly onto an isolated colony on the culture plate, and observe the color change on the colonies for up to 60 seconds. If colonies turn purple or dark blue color within 10 to 60 seconds, the test is positive. And colonies with no change color mean the test is negative. Helpful tips. Do not use a nichrome loop. Nichrome loops, a nickel-chromium alloy, may oxidize and lead to false positive results. The other one is use fresh cultures. Use 18 to 24-hour cultures so that the bacterial enzyme activity is at its peak. And the last one is accurate timing. The color change should be observed at the specified time. Any color change after the specified time, usually 60 seconds, should be ignored. Importance of the test. The oxidase test is particularly useful for distinguishing between the Enterobacteriaceae family, oxidase-negative, and other gram-negative bacteria such as Pseudomonas, Neisseria, Aeromonas, Vibrio, and Campylobacter, oxidase-positive.